So thank you for the invite to join this group today. It's been a much more interesting Friday afternoon than I usually have at work, so I've really enjoyed it so far. Thanks for everyone's contribution. So I, by training, I'm a biochemist and want to talk a little bit more about the biochemistry of collagen. The article, the review that we wrote is Collagen Through the Life Stages and its importance, from youth to adulthood, changes with menopause, etc. But actually, just for the next 15 minutes, I want to concentrate on collagen, the importance of collagen and its formation in the skin for us, obviously the fibroblasts. Other groups might be more interested in chondrocytes for the joint or osteoblasts for bone, etc. That's a wonderful thing about collagen. It's very ubiquitous. It's very important. It's a very highly conserved protein in evolutionary terms. So I just want to talk a little bit about the importance of collagen, how it's made, and obviously because we're Minerva Research and we use hydrolyzed collagen products, also talk a little bit about the strategy we use to try and promote new collagen synthesis in skin or joint would be very similar, etc. Oh, so sorry, this was supposed to come up as one slide. There we go, a little bit better. So we make collagen-based liquid supplements, and that is usually hydrolyzed collagen, can then work through peptides or free amino acids to stimulate fibroblasts to build synthesized new collagen fibers. So that's the first and most important mechanism of action of hydrolyzed collagen. So that's stimulating new collagen synthesis in skin or joint. For that to be successful, it does require specific vitamins and minerals that are required for both the synthesis of collagen and the assembly in the extracellular matrix. So I think one of the weaknesses with some of the approaches out there is that people just talk about synthesis of collagen, making new collagen, but actually as a protein, it has to be assembled in the right three-dimensional conformation in combination with elastin, glycosaminoglycans, proteoglycans. It has to be assembled into this extracellular matrix, and it's a very carefully orchestrated event or series of events, and a very complex series of events. And knowing those steps in the biochemical details is what will help us be more efficient at promoting that process to happen in skin. I'm not going to talk about them today, but I think the other participants have talked about the importance of antioxidants. We heard Dr. Burke talking about UV damage, oxidative stress. Antioxidants are important for slowing down the natural degradation of collagen and elastin fibers, protecting the extracellular matrix. And we've seen some fantastic images of what happens when we fail to do that, how aging, wrinkles, and just the facial in particular damage that occurs. And of course, the other part that is important to that is the support ingredients, the botanicals and the bioactives that support that. So always it's about as many interventions, minerals, vitamins, hydrolyzed collagen, peptides. The more targeted those interventions, the better. So I just wanted to start by saying a little bit about native collagen versus hydrolyzed collagen. I don't know, but I always get the impression there's still a lot of confusion or perhaps misconceptions about what is native collagen compared to hydrolyzed collagen. So native collagen is the long chain triple helix structure that is in extracellular matrix in the body. It's about 1,000 amino acids in each chain. There are 3 chains tightly wound together to make a robust protein that is then assembled into a super molecular structure. It's very high molecular weight protein, not easy to digest or absorb. This is where hydrolyzed collagen comes in. Hydrolyzed collagen is basically an industrial process by which you take collagen, usually from skin, from bovine or from fish, for example, and they just denature it and hydrolyze it, usually with pH, acid, and with enzymes. And it's mimicking the digestive process. So hydrolyzed collagen just mimics what we would do in the gut. Breaks up the long, high molecular weight protein into short chain peptides, which are low molecular weight, and therefore soluble. And that's one of the main attributes of it. It's soluble, so it's easy to formulate, and it's compatible in a solution that we can then present to the consumer. And then it's easily absorbed and distributed within the gut and the bloodstream. So just a little explanation of that. Collagen from bone, from cartilage, from skin, This will be the 3 strands woven together, and then they're assembled into a large supramolecular structure. That's, basically skin, tendon, joints, and bone. And through heating, the first step, usually cooking, we denature the structure and we start breaking it down and releasing the smaller components. First, we release the triple helices, and then we break them apart. And in doing that, we go from collagen, which is a molecular weight of over 300 to 400 kilodaltons, to gelatin. So that's the denatured cooked form of hydrogen, which is probably the first step in terms of breaking up the collagen structure. For gelatins, it's a large spread of molecular weights. It can be anything quoted from 50 to 200 kilodaltons, because it depends on how much you denature and how you treat. It can also be acid hydrolysis and enzymatic hydrolysis, which will give us much smaller molecular weight peptides. And these are the hydrolyzed collagen that are in so many products, including our own. They're usually around 2 to 5 kilodaltons. There's still quite a few amino acids, but they are much, much smaller and therefore soluble. And then when we ingest those through a supplement, they are further hydrolyzed in the small intestine to collagen di and tripeptides and free amino acids. So as I said, the collagen forms that we will have in a product are basically an industrial process that mimics digestion, because that's how we ultimately want to get them into the bloodstream, the way the digestive system will. So it is important to notice that only amino acids, dipeptides, and tripeptides are absorbed into the bloodstream, specifically through amino acid transporters and peptide-1 transporters. It's a very active and carefully coordinated process. So we know that specifically these amino acids are taken up. As in any biological system, there are exceptions to the rule. There are medical conditions with chronic inflammation where larger peptides can get access to the gut and to the bloodstream, but they're the exception to the rule. Under normal circumstances, this is how the peptides are presented into the bloodstream. So from the bloodstream, then throughout the body, they will diffuse basically, or perfuse, I should say, excuse me, and then gain access to the, by leaking out of the capillaries, gain access to the fibroblasts in skin in our case. The fibroblasts are embedded in an extracellular matrix. And the matrix itself is really important to the stimulation of the fibroblasts. So fibroblasts respond first and foremost to mechanotransduction. A fibroblast in vitro, in a test tube, if it doesn't have a support matrix, it's not very active. So we know if we give it a high tension support matrix, physically the mechanotransduction will stimulate the fibroblast. And that's one of the properties you lose with aging. As our skin sags, the extracellular matrix is weak, those mechanical forces are weak, and the fibroblast becomes less active. It's also important for ligand-induced activation. So peptides, or it could be drugs, obviously, as well. Ligands can also get from the bloodstream to the fibroblast and stimulate to increase activity, not just of collagen, but of also elastin, GAGs, proteoglycans. And of course, finally, there's also autocrine, paracrine, and endocrine stimulation. So hormonal type factors, for example, and we all know the decrease of estrogen post-menopause will be related to a decrease in collagen production. So lots of factors that can stimulate the fibroblasts and that we need to think about when we're trying to induce the fibroblasts to make new collagen fibers. And that's just shown in our diagram on the right. It can be activated by physical stimuli and or by ligands, which are the di and tripeptides from the hydrolyzed collagen. These stimulate the fibroblasts to produce new collagen proteins, and then very importantly to assemble them in the extracellular matrix. And that was the key message in the paper we wrote that I wanted to emphasize and want to talk a little bit more detail today. So I just want to spend a little bit of time talking about this because I kind of marvel at the biochemistry and the biology of systems. If this was something like building a space station, we launch the shuttle on Earth, put modules in space, and then we assemble them into the International Space Station. And the fibroblast does something similar, I should say. It makes the collagen fibers, but they're kind of big, so it has to secrete them into the extracellular matrix and then further processing to assemble them remotely. And it's an amazing feat of biochemistry. So I'd just like to spend a few minutes going through that. So basically we start with the endoplasmic reticulum in the cell where the protein synthesis is made. And you will first produce a single strand of collagen, which will be about just over 1,000 amino acids. Through lysine and proline hydroxylation, you'll add hydroxyl groups to specific points along the backbone. It's not random, it's very well coordinated in specific activity. And these are important because these will really dictate how the collagen fiber is produced and assembled. Some of them can be glycosylated, so they will have sugar moieties attached, and they're very important. And eventually you have 3 of these units, which are basically attached and wound together to form a triple helix. And it's a very tight triple helix. It's a very stable triple helix. Even the amino acids, the glycine, the proline, hydroxyproline, form a very tightly wound, very stable chain, and then a tightly wound, very stable triple chain, basically. And this has produced a single unit and then is shuttled along the Golgi apparatus out of the fibroblast cell and into the extracellular matrix. When this unit is shuttled into the extracellular matrix, the first thing the tissue will do is cleave the N and the C terminal peptides. And that releases then our tropocollagen, which is associated with a triple helix with the telepeptide ends. And as I said, these very important hydroxyl structures and glycosylated structures. This unit then needs to be assembled, and it's assembled then by, very specifically cross-links that are occurring at chemistry at the lysine residue. So the lysine residue at the terminals of each of these peptides has to be oxidatively deaminated, forms a free radical, and then has a condensation reaction with a hydroxylysine on the adjacent strand. And through doing this repeatedly, we cause all these cross-links to be formed, and they are very specific. Cross-linking, if there's too much and it's not very specific, can damage the collagen. And in fact, that's what happens later in life through ultraviolet radiation and through advanced glycation end products. For example, we have lots of non-specific cross-links. So it's very carefully orchestrated so that the correct chemistry produces the correct condensations to link in a very coordinated way and produce then what is fibril for skin and a slightly different fibril for joints. So for example, the chemistry of the cross-links will be different in cartilage than it is in skin. And that's just a way of making different collagens and structuring them a little bit different. The body just uses different chemistry to achieve the different three-dimensional structures of collagen fibers that it produces. So these lysine residues have been modified. They're ready for use in the extracellular matrix, and the chemistry is what forms the cross-links and allows those adjacent units to assemble into the large bundles we know as the collagen fibers that create the extracellular matrix that dictates skin elasticity and the scaffolding of skin. And then just very important for us, knowing the biochemistry in detail is important. I think everybody, even school children, know that vitamin C is important. And if we don't have vitamin C, we have scurvy. And of course, that is because vitamin C is required specifically as a cofactor for the hydroxylation, for the prolyl 4 hydroxylase enzyme that causes these early reactions. It's also a cofactor for a lysyl hydroxylase assay. So without vitamin C, we cannot make these collagen fibers. But equally important, once the collagen fibers are synthesized and then transported into the extracellular matrix, we need copper and vitamin B6 to drive, but they're required as cofactors for the deamination and the cross-linking condensation reactions that generate the cross-links. So knowing more about the biochemistry, about how this fibril is assembled, not just synthesized, I think is really, really important. And it's helping us to make more targeted and more effective way of developing the supplements. Because supplements, I think, have their forms, have their uses, but also have their weaknesses. And we're trying to use the biochemistry to drive that a little bit better. So just in summary, to say, there are 28 collagens. I'm only talking about collagen type 1 here today. It's the classic type collagen. It's the one I think everybody knows or probably understands if you talk about skin and extracellular matrix. But of 28, 5 or so are fibrillar collagens, and the others have varied functions role. I think in the last decade, the interest in collagen as an extracellular matrix component has increased. And I think that is due to a renewal based on collagen supplements suddenly becoming popular in the media, etc. And the ability then to stimulate collagen synthesis and extracellular matrix production, whether it's fibroblasts in the skin, like I say, chondrocyte in joint, is key. I just wanted to finish with perspective and an opinion really from myself. I think that for me is a very nice story. It's a very nice picture of how we can help the surgical interventions by use of supplements. So, after surgical interventions, and we've done some work with surgeons in Italy, in particularly promoting and trying to tell people to use these kind of supplements post-surgery to help recovery. But one of the most difficult factors still to overcome is self-senescence. And I think this is particularly important in aging, where after so many cycles, particularly through youth and through growth and development, the cell becomes less responsive and becomes difficult to stimulate that biochemistry and that new collagen production. So I think that's where we, in research terms, are really trying to provide future focus and future benefits. With that, I'd just like to thank the editorial team for the chance to talk today. Thank you.
Thank you. Thanks for Dr. Reilly's sharing. Let's enter the discussion session. Do you have any questions for Dr. Reilly?
Yeah.
Yeah. I have a question. And I think from your wonderful speech, we got to know the process of the collagen synthesis and degradation. Of course, because we are practice surgeons, we want to use the collagen or collagen products to do some anti-aging treatment. Of course, the natural collagen is too expensive. However, the gelatin or the hydrolyzed collagen or the peptide, I think they're much cheaper. And my question is, after injection, for example, with the gelatin or hydrolyzed collagen, what's the fate of this collagen? They will continuously be degraded into the amino acids and then reabsorbed into the bloodstream, or they will be reassembled by the fibroblast into the natural collagen, I mean, with the long and the thick bundles.
So I think both of those things happen. I think the fibroblast has to make from amino acids a new collagen fiber. So and the peptide just stimulate the fibroblast to do that. So you're providing both the peptide to stimulate the fibroblast, that's the first mechanism, but you also need to supplement with the amino acids. And in the digestion, the collagen or gelatin or hydrolyzed collagen all have to be degraded right down to amino acids and ditripeptides. Under normal healthy circumstances, nothing bigger than that can really effectively get into the bloodstream. There are clinical chronic inflammation conditions, but under normal circumstances, that's as large as you can transport through the bloodstream to the fibroblast. So you're providing amino acids, which are the building blocks of new collagen, but you're also providing ditripeptides, which will stimulate the fibroblast to produce new collagen. And I think both mechanisms are, in my opinion, equally important.
So you mean the long chain peptide, such as the gelatin, can be directly reused by its fibroblasts to make the collagen, right?
No, it's too big. It's too large for the collagen, for the fibroblast to absorb, to transport. So one of the most important things in biology, whether it's from the gut into the bloodstream or the bloodstream to the fibroblast, is the size of the molecules we can transport. So it doesn't matter whether it's proteins, sugars, fats, or even minerals and vitamins. We have to break everything down to the smallest absorbable unit and then reassemble. And it's a common process. It doesn't matter whether it's proteins or sugars or fat. We break everything down and then we reassemble via fibroblasts or other cells throughout the body. So I think that's just how we handle proteins, carbohydrates, fats. We break them down because they're too big to transport.
So the supplementation or the addition of the gelatin or the hydrolyzed collagen will be beneficial for the fibroblasts to make collagen, right?
Yes, in the long run, yes.
Okay, thank you.
Thank you. Sorry, Dr. Burke, you're on mute.
Okay.
Still on mute.
I mean,
There
I
we
think
go. Yes. Nope.
now I'm okay.
Okay.
Yes.
Okay. So we found that just implanting collagen itself and most other implants, as it turns out, but especially collagen, brings fibroblasts to the site, and fibroblasts then synthesize new collagen. So partially from the substrate as the broken down pieces, as you said, but just the implant itself stimulates the body to bring in fibroblasts. And also, we did some very interesting experiments very early. This is one of my favorite experiments, that we took fibroblasts from neonates and fibroblasts from people over 90 years old. And we found, obviously, that the fibroblasts from the older people proliferated less well and synthesized a lot less collagen. But then when we added vitamin C, 5% even, or 10% to the media, the elderly fibroblasts equaled the neonatal fibroblasts in proliferation and produced even more collagen than the neonatal ones. So just adding a lot of extra vitamin C stimulated elderly collagen, elderly fibroblasts to produce more collagen.
The response, yes, very interesting.
Yeah. Yes.